Hello guys, here I am to talk about the novel coronavirus again. Now it gets a new name, SARS coronavirus 2 by ICTV, International Committee on Taxonomy of Viruses. It causes the disease novel coronavirus pneumonia, which is now coronavirus disease 19. After checking and reading over 70 published papers, I found more evidences new and old to overthrow the conspiracy of virus leakage. Now, here's the thing. The novel coronavirus pneumonia is not only take place in China, but also presents a very rapid growth rate of infection in Japan, South Korea, Italy, and other countries globally. Even though the mortality of NCP, novel coronavirus pneumonia, is about 2.3% till now when compared with 9.6% of SARS and 34.4% of MERS, as reported by WHO. It still causes a great casualty due to its highly contagious ability. To know why the new virus is that highly contagious, we need to know about the S protein of the coronavirus first. As we've mentioned in the last video, the S protein is the key for the virus to invade into the host cells. Now it's spotted by people who believe in virus leakage conspiracy and the scientists at the meantime. The S protein of coronavirus has two parts, S1 and S2. There are also structures that anchor the S protein to the capsid membrane lipid bilayer, which is the same structure with the cell membrane. For the S2 region, it includes the HR2 region and HR1 region. HR is short for peptide repeat. S1 region contains the RBD, the receptor binding domain, and FP, the fusion peptide. According to the name of RBD, it has the function to recognize the receptor ACE2 on the host cell membrane. Once the virus binds to the ACE2 by the RBD, the S2 region will start to work. HR1 and HR2 will interact to help the capsid membrane fuse with the host cell membrane. Then, the virus will succeed in invasion. The very recent research in 2020 February by Etienne Decrolli and Jason McClellan pointed out that there is a special site called the furin-like cleavage site inserted into the area between S1 and S2. And this structure can facilitate the entry process of coronavirus into the host cell. Can you find this structure in the SARS coronavirus? No, but you can find this in the highly pathogenic flu virus commonly. This special site makes the new coronavirus more contagious than the SARS. Further analysis of amino acid sequence indicated that the novel coronavirus and the SARS coronavirus are 100% identity in the HR2 area, which is highly conserved across species of viruses. However, there is only 62% identity of HR1 core amino acid between novel coronavirus and SARS coronavirus, while direct ancestors of SARS coronavirus RS-SHC014 and WIV1 and RS-3367 are 100% identity with the SARS coronavirus. Now you might wonder, how about the rest region of S protein, RBD? When the scientists try to use antibody like M396 and CR3014 that can neutralize SARS coronavirus, it turns out that they can't bind to the RBD of novel coronavirus. This suggests that amino acid sequence identity between RBD region and the novel coronavirus and the SARS coronavirus is low. Further research showed that S protein of the novel coronavirus is 98% identity from the bad SARS-like coronavirus RATG13. Now let's conclude. First, the furin-like cleavage site between S1 and S2 only present in novel coronavirus, instead of other SARS coronavirus or bad SARS-like coronavirus. Second, identity of amino acid sequence of HR1 core is low when compared to the novel coronavirus with SARS coronavirus. Third, RBD region shows a difference between that of the SARS coronavirus and the novel coronavirus. Fourth, spike protein of novel coronavirus is more like a bad SARS-like coronavirus RATG13 with an over 96% whole genome identity. And fifth, the ACE2 is a highly conserved protein in human cell and widely distributed across different organs. How could any country spread the virus to a specific country? It will spread globally. Look at Italy, Japan, South Korea, and America now. With those reasons above, trust me, it's unlikely to have this epidemic disaster due to virus leakage. So, according to the different function of the areas in the S protein, can you see why we need to study viruses? Here, in this picture, you can find the receptor binding domain. The receptor binding domain may give us some clue of developing vaccine or antibody to block the process of recognition for the virus when they try to get invade into the cell. And for the HR1 region, we may find some fusion inhibitor to stop the viral entrance process. We need to study about viruses is because there's still too many questions unsolved. As we've discussed previously, RATG13 has over 96% identity from the whole genome level with the novel coronavirus. The scientists found this virus in a horseshoe bat. Then, how could the RATG13, the potential candidate virus, turn into SARS coronavirus 2, which is the novel coronavirus? Two possible ways: recombination or mutation. The recombination of gene among different bat species is still need to be further researched. The picture that you can see here shows about the horseshoe bat. It has a special nose structure. As the natural reservoir of many coronavirus includes the RATG13, how the mutation take place when the virus transmits to human still got a lot of questions to be answered. So we still need to study about the viruses. Even though you are not doing experiments on the viruses never cease mutating. Human never cease the interaction with the other viral host animals. No matter what, viruses are always several steps ahead of human, and the study of how viruses invade into cells using pseudotyping viruses or other method, and the study of how viruses take control of cells are ways for us to lessen the steps between viruses and human. Some people question that, is there still a chance for the virus to leak out of the lab? Yes, it still has a minor possibility. Should we worry about it? We should, but we shouldn't panic or hate everything associated with virus. Except for the strict laws and regulations, we also have the supervision from people and morality. So, what's the most important thing of being human? Happy? No, it's having conscience. That's the most important thing, and that should be the core of education, of family education, of school education, and of social education. That is all I want to talk in this video. Thank you so much for watching.